Sucrose and sucrase. Sucrose is a disaccharide consisting of two monosaccharides, fructose and glucose. As our cells can only absorb monosaccharides, sucrose must be broken down into its two fundamental products. To do this, our small intestines are coated with multiple types of enzymes by the millions. The ones that specifically deal with sucrose molecules are called sucrase. Sucrose cannot be broken down by any other enzymes other than sucrase, as the sucrase active site is the only one that supports sucrose. In this case, sucrase will act as the enzyme and the sucrose as the substrate. When the sucrose collides into the sucrase active site, an enzyme substrate complex is formed. The enzyme takes in water to weaken the bonds in the sucrose, thus classifying as a hydrolysis reaction. In this reaction, the enzyme would be a catalyst, cutting down the energy requirement to break the bonds in sucrose. Without a catalyst, higher amounts of energy would be required to break these bonds. However, with catalysts, although the process would take the same amount of time, they cut down the energy requirement significantly. Once the bonds in the sucrose are broken, the X-disaccharide is now two individual monosaccharides and can finally be absorbed by our cells. To prevent sucrose from entering the active site, we would bring in inhibitors. We will talk about competitive inhibitors. Now, competitive inhibitors interact with the enzymes in a way that blocks off the enzyme from the substrate by binding itself to the active site. This way, the substrate bounces off the enzyme and goes right past the digestive system, thus incapable of being absorbed. Thank you.